Okay, so this is part of the A-level AQA syllabus, the specification reference as part of the DNA and RNA structure, where it says that the relative simplicity of RNA led many scientists to doubt that it carried the genetic code. So this is kind of how science works. So unraveling the role of DNA. So DNA is a hereditary material which passes on genetic information. At one time, some scientists thought proteins did this. How DNA rather than proteins were proved to be the hereditary material is an example of how science works. DNA versus protein. Chromosomes are only visible during cell division, and scientists focused on them as hereditary material. Chromosomes are made of protein and DNA. To produce variety, material must be diverse, so each organism has its own specific type. And as proteins have chemical diversity, they thought that they were the hereditary material, and the enzymes and antibodies showed specificity. In contrast, DNA had few components, and they thought it was too simple. But some scientists were not convinced and set about to prove it was DNA. So evidence that DNA is the hereditary material. So observations and knowledge to form of the hypothesis is where they started. They made predictions for the outcome of investigations. They repeated experiments to gain experimental evidence to accept or reject a hypothesis. And by the 20th century, they had evidence that DNA was the hereditary material. It is present in chromosomes, it's stable, and the quantity halved in egg or sperm, but not in body cells. Proving that DNA is the hereditary material. So experiments using mice and bacterium causes pneumonia. And there are two forms. There's a safe form, which is the R strain, and there's a harmful form which actually causes the pneumonia, which is the S strain. So mice were separately injected with living safe form and dead harmful form. Both groups remained healthy. When the mice was injected with both forms, however, they developed pneumonia. Living bacteria were isolated from mice with pneumonia, and there were three possible explanations. Either it was experimental error, could be that the living safe form mutated into a harmful form, which is very unlikely, but probably the pneumonia caused by toxins, and harmful bacteria has information on how to make the toxin, but being dead it can't do so. So the safe form can make the toxin, but lacks the information to do so. Information on how to make the toxin is transferred from the harmful to the safe form, which then produced it. But then the third explanation, which is definitely worthy of more investigation, is that the living harmful bacteria in mice with pneumonia was collected and the substances were isolated from the bacteria, and each substance added to suspensions of living safe bacteria to see if it would transform them into harmful form. And only the substance produced this transformation was DNA. When the enzyme that breaks down DNA is added, ability to carry out the transformation stopped. They then did other experiments. So viruses infect bacteria, making them make more viruses. And because viruses are made up of just protein and DNA, it must possess instructions that bacteria need to make new viruses. Protein and DNA in viruses can be labelled with the radioactive element. One sample of bacteria infected by viruses with radioactive protein and another with viruses with radioactive DNA. Later stage viruses and bacteria in both samples were separated from one another, and only the sample with bacteria infected by viruses labelled with radioactive DNA showed radioactivity. So how science works. So evidence that DNA material provided bacteria with genetic information needed to make viruses. DNA can be passed from one bacterium to another, and further proof is that electron microscope studies trace movement of DNA from viruses into bacterial cells. So they started off with observations which led to a theory. They had a hypothesis to explain the observations. They then did experiments designed using methods to test the hypothesis. The experiments were repeated to make it reliable. The results were analysed and interpreted to accept or reject the hypothesis. The experimental techniques were critically reviewed by other scientists. Experiments refined to lead theory to be accepted or modified, and the knowledge was used in other aspects of science like medicine. The benefits and risks of discoveries were analysed by society, and the ethics of how the experiments were carried out were considered.